Hello everyone. In the last video on this chapter, we discussed about inheritance of one gene, inheritance of two genes, back cross, test cross and its significance, principles of Mendel's or first, second and third law of Mendel. This video mainly deal with concept of dominance, other patterns of inheritance, we simply call it as non-Mendelian inheritance, in which incomplete and codominance. The concept of dominance. As we all know, gene is the unit of inheritance. Gene is the unit of inheritance. Every gene consists of the information to express a particular character. Every gene consists of the information that express a particular character. In a diploid organism, there are two copies of each gene. In diploid organism, there are two copies of each gene. That is nothing but a pair of alleles. In homozygous condition, there are identical alleles. In heterozygous, there is non-identical alleles. That is, one dominant, another one is recessive. The normal allele or unmodified or functioning allele of a gene produces normal enzyme that is needed for the transformation of substrate. Normal or unmodified or functioning allele of a gene produces normal enzyme that is needed for the transformation of substrate. We call it as a dominant allele. Whereas another may be different due to some change that it has undergone which modify the information that particular allele contain. So, another may be different due to some change that it undergone that modifies the information that particular allele contains, that we call it as modified allele, and it is responsible for production of a normal or less efficient enzyme or a non-functional enzyme or no enzyme at all. This modified allele is responsible for production of the normal or less efficient enzyme or non-functional enzyme or no enzyme at all. In the first case, that means the normal or less efficient enzyme. In the first case, the modified allele will produce the same phenotype like unmodified allele. The modified allele will produce the same phenotype like unmodified allele. That means the modified allele is equivalent to unmodified allele. It is equivalent to unmodified allele. And it becomes dominant But in second and third case, second case is production of non-functional enzyme. Third, no enzyme at all. In a second and third case, the phenotype will depend only on the functioning of unmodified allele. The phenotype will be dependent only on the functioning of unmodified allele. Here the modified allele become recessive. Here the modified allele become recessive. So, the recessive trait is due to non-functional enzyme or no enzyme is produced. Normal gene is responsible for, or normal allele is nothing but the dominant allele. The modified allele means it is a recessive allele. This recessive allele sometimes act as a dominant when it is produced. From this concept of dominance, we understood that this recessive character is only due to production of non-functional enzyme and no enzyme at all. Production of non-functional enzyme and no enzyme is produced. Then only the recessive trait is expressed or recessive character is expressed. Understood? Here the normal or unmodified allele is nothing but dominant allele. Modified allele is nothing but recessive allele. Sometimes this recessive allele also acts as dominant when it is produces normal or less efficient enzyme. Okay, let us try to understand about concept of dominance. First, we know that gene is the unit of inheritance. Gene is the unit of inheritance. Unit of inheritance. Clear? This gene carry the information. Carry the information. Carry the information for the expression of a character. For the expression of a character. So, gene is the unit of inheritance which carry the information for the expression of a character. Which carry the information for the expression of a character. Clear? In diploid organism. In diploid organism, there are two alleles. In diploid organism, there are two alleles for a single gene. For a gene. In diploid organism, there are two alleles for a gene. In homozygous condition. In homozygous condition, there are identical alleles for a gene. Identical alleles for a gene. In heterozygous condition, there are non-identical alleles for a gene. There are non-identical alleles for a gene in which one is dominant, another one is recessive. One is dominant, another one is recessive. Okay, gene is the unit of inheritance which carry the information for expression of a character. In diploid organism, there are two alleles for a gene. In homozygous condition, identical alleles for a gene. In heterozygous condition, non-identical alleles for a gene in which one is dominant, another one is recessive. This is clear for you? Okay. Next, moving to consider this dominant and recessive. This dominant and recessive gene as this is a functional gene. This is a functional gene or functional allele or unmodified allele or normal allele. And this recessive as a modified allele. Consider dominant allele as a unmodified or normal or functioning allele. Whereas this recessive one is a modified allele. Recessive one is the modified allele. First try to understand about this dominant or unmodified allele. Consider this normal allele produces normal enzyme or functional enzyme. Normal enzyme or functional enzyme. This enzyme is needed for transformation of a substrate into a character or a phenotype. Transformation of a substrate into a character or a phenotype. So, dominant or we call it as unmodified, normal or functioning allele responsible for the production of normal enzyme that needed for the transformation of a substrate into a phenotype. And this modified allele is modified or changed its character due to some change it undergone. It modified its character due to it undergone some change. This modified allele either responsible for production of a functioning enzyme or functional enzyme, production of functional enzyme, or little bit less efficient enzyme, or it may produce as non-functional enzyme, or no enzyme at all. So this modified allele is modified or get changed due to some change it is undergone. As a result, it may responsible for production of functional enzyme or little bit less efficient enzyme, and non-functional enzyme or no enzyme at all. In a first case, this normal enzyme or less efficient enzyme produces or required for transformation of a substrate into normal phenotype. In this case, this modified allele is equal to this unmodified allele. This modified allele equivalent to unmodified allele. In this condition, it acts as dominant one. It also acts as dominant one. In this condition, this recessive allele acts as dominant one. It is equivalent to this unmodified allele. But in second and third case is production of non-functional enzyme. Third case is enzymes are not produced. In these two cases, what happened? There is no production of enzyme or produced enzyme is non-functional. In that case, there is no transformation of a substrate. Therefore, no phenotype is produced. Phenotype is not produced. No transformation of substrate, therefore phenotype is not produced. In that case, the phenotype is only depend on this dominant allele. In that case, the phenotype is depend on the dominant allele or normal allele. So, this recessive character is mainly depend on the production of non-functional or production of no enzyme. Production of non-functional or production of no enzyme is responsible for the recessive character. Understood? In this case, this recessive gene or modified gene failed to express itself. But in first case, it expressed itself. Therefore, it is equivalent to this dominant gene. Understood this concept of dominance? So, in heterozygous condition, there is one dominant, another one is recessive. One which express itself is called as dominant. One which fail to express itself is called as recessive. Sometimes this recessive also expressed when it produces a functional enzyme that is equivalent to dominant or normal allele. So next is other patterns of inheritance. We call them as non-Mendelian inheritance because this inheritance do not obey the Mendel principles. They are deviated from Mendel's principle. So they are called as non-Mendelian or other patterns of inheritance. For your study, there are 5 different non-Mendelian inheritance. They are incomplete dominance, codominance, multiple allelism, polygenic inheritance and pleiotropism. Incomplete dominance, codominance, multiple allelism, polygenic inheritance and pleiotropism. But this video only deal with incomplete dominance and codominance. So first discuss about the incomplete dominance. Incomplete dominance is a inheritance in which a heterozygous offspring shows the intermediate character between two parental characters. The heterozygous offspring shows the intermediate character between two parental characters. For example, flower color of a snapdragon or dog flower plant and flower color in mirabilis jalapa or 4 o'clock plant. The flower color in snapdragon or flower color in mirabilis jalapa. In your textbook, they have given the example of flower color in snapdragon. When a pure red flower plant is crossed with a pure white flower plant, in F1 generation, all pink color flower plant is obtained. When a pure red flower plant is crossed with a pure white flower plant, in F1 generation, all pink flower plant is obtained. When F1 is selfed or F1 hybrid were crossed, in F2 generation, both phenotypic and genotypic ratio is same. Both phenotypic and genotypic ratio is same. That is 1:2:1. In phenotypic ratio, 1 red, 2 pink and 1 white. In genotypic ratio, 1 capital R, 2 capital R small r and 1 small r. The phenotypic ratio is deviated from actual ratio 3:1. This means that R was not completely dominant over small r. Capital R was not completely dominant over small r. In this flow diagram, you can observe the red flower plant, that is capital R, white flower plant, small r crossed. In F1 generation, pink color flower is obtained. That means the heterozygous offspring shows the intermediate character between two parental characteristics. Therefore, it is an incomplete dominance. In F2 generation, the phenotypic and genotypic ratio is 1:2:1. 1:2:1. So in incomplete dominance, both alleles of a character expressed their character in F1 generation, so that F1 individual has an intermediate character between the parents. So F1 has an intermediate character between the parents, and the hybrid has a new phenotype which has no new gene. Hybrid has a new phenotype which has no new gene. There is a blending of this character in hybrid. There is a blending of character or mixing of character in this hybrid. But remember, the reappearance of pure parental character in F2 generation shows that there is no mixing of genes. There is no mixing of genes. But in hybrid, both the characters are blend. But there is no mixing of genes. Okay. Here, the gene red color is not completely dominant over white color. The gene red is not completely dominant over the gene white. Hence, the intermediate character, that is pink color, is expressed. This cross also shows that there is no specific gene for pink color. There is no specific gene for pink color. Next example for incomplete dominance is inheritance of flower color in snapdragon plant. Inheritance of flower color in snapdragon plant, and it was studied by Carl Correns. He crossed a pure red and a pure white color flowering plants. Pure red and pure white flowering plants. Okay, these are the gametes, and this is F1 generation. F1 generation, he got all pink color flower plants. But according to Mendel, when dominant gene is present along with recessive gene, dominant gene express itself and the flower color become red. But here it is deviated from Mendel principle and a new color obtained, that is pink color flower is obtained, which is the intermediate character between these two characters. This indicates this capital R is dominant over small r. Capital R is dominant over small r. Next he crossed this F1. He self this F1. These are the gametes in F1 generation. So F2 where capital R, capital R, capital R, small r, capital R, capital R, small r, small r. This is red one, this become pink, this is pink and this is white. That means out of 4 plants he got 1 red, 1 white and 2 pink. The phenotypic ratio is 1:2:1 and genotypic ratio also same, that also 1:2:1. So this phenotypic ratio is deviated from the Mendel actual monohybrid ratio, that is a 3:1. So it is deviated from this 3:1 ratio. New ratio is formed, that is 1:2:1. This indicate this capital R is incompletely dominant over small r. Capital R is incompletely dominant over small r. Remember one thing, these genes are not mixed. These genes or these alleles are not mixed. Why these are not mixed? Because in F2 generation, he got a pure red and pure white color flowering plants. This indicates genes are not mixed, but characters are mixed in hybrid. Characters are blend in hybrid, but genes are not mixed. They are free from each other. So in F2 generation, he obtained pure red and white. That means pure parental character he obtained in F2 generation. Okay. This is for the incomplete dominance. Understood? So next is codominance. Codominance is the inheritance in which both alleles of a gene are expressed in a hybrid. For example, AB blood group in human. ABO blood group is controlled by capital gene I. ABO blood group is nothing but in human the blood group is called as ABO blood group. It includes 4 different types of blood group, that is A, B, AB and O. This is controlled by capital gene I. This gene is responsible for production of sugar polymers or antigens that protrude from the plasma membrane of RBC. This gene has 3 alleles, IA, IB and small I or I0. IA and IB produces a slightly different form of sugar, while the small I does not produce any sugar. That means IA and IB produces slightly different antigen, whereas small I does not produce any antigen. IA and IB completely dominant over small I. IA and IB is completely dominant over small I, whereas IA and IB present together, they both express their own type of sugar. This is due to codominance. So ABO blood group is controlled by capital gene I, which has 3 alleles, IA, IB and small I. This gene I is responsible for production of antigen which present on the plasma membrane of RBC as a small protrude. Okay. IA and IB are dominant over small I. IA and IB produces slightly different antigens. IA responsible for production of antigen A, IB is responsible for production of antigen B, whereas small I does not produces any antigen. When IA and IB present together, both produces antigen. That means IA and IB present together, antigen A and antigen B is produced and blood type is AB. This is due to codominance. In this chart, we observe that the alleles from the parent 1, alleles from the parent 2, and then genotype in the offspring and a blood type or phenotype in the offspring. In clear observation, we found that there are 6 different types of genotypes and 4 different types of phenotype, which are the A, B, AB and O are the phenotype, which are the genotype IA small I for A phenotype. Next, IB and small I for phenotype B. IB for phenotype AB and small I for phenotype O. So, when IA present with small IA is expressed and blood group become A. Similarly, IB is present with small I, then the blood group become B. Only O blood group occurs when both the recessive allele come together, that indicate IA and IB are completely dominant over small I, and for AB blood group, IA and IB present together, then blood group become AB. Here, the blending of character not occurs in hybrid. The blending of character not occurs in a hybrid. Understood? Next, codominance. For codominance, example is the AB blood group in human. First, try to understand ABO blood group. In human, the blood group is commonly called as ABO, which include 4 kinds of blood groups. They are A, B, AB and O. A, B, AB and O. The production of this ABO blood group is controlled by a gene called I. Capital I. This gene is responsible for production of antigen, or simply we call it as sugar polymers, which present on a membrane of RBC, as a protrude or outgrowth. So gene I is responsible for production of antigen or sugar polymers that present on the surface of or membrane of RBC as a outgrowth or protrude. This gene I has 3 alleles. They are IA, IB and small I or I naught. Gene I has 3 alleles, IA, IB, small I or I naught. This IA and IB produces antigen which is slightly different from each other. This IA is responsible for production of antigen A. Okay, this IB is responsible for production of antigen B. But the small I or I naught do not produce any antigen. No antigens are produced. So, IA and IB produces slightly different form of sugar polymers. That is, antigen A is produced by IA and antigen B is produced by IB. But small I do not produce any antigen. Remember, IA and IB, these both are completely dominant over small I. IA and IB are completely dominant over small I. That means IA present with small I, then antigen A is produced and blood group become A. When IB is present with small I, then antigen B is produced and blood group become B. IA and IB present together. IA and IB present together. Both antigen A and antigen B is produced. Both antigen A and antigen B is produced and blood group become AB. This is due to codominance. When IA and IB present together, when IA and IB present together, both antigen A and B is produced and blood group become AB. Clear? Suppose this II, small I, alleles are come together. They are present in homozygous condition. Then there is no production of antigen and blood group become O. Understood? That means IA is responsible for blood group A, IB is responsible for blood group B, small I is responsible for blood group O, and both IA and IB is responsible for blood group AB. Understood? Clear for you? This is the end of the slide. So thank you everyone for your attention.